I think we could start.
I think so as well. So I'll let you do the introduction. So I'm
Sure.
the camera as well.
Okay. So, well, hi everyone. So Ian is a PhD student in the area of XI, so explainable AI. He's also at his last year. Am I correct, Ian?
Yeah, like I'll probably defend within the month or a little bit more. Just right at the end.
Okay, so it's and you're from the university. You're in Brazil.
Yeah, in Rio.
Yeah, the Catholic University from Rio de Janeiro. So he's calling us from Brazil, and that's the reason why we moved our lunch to 4 p.m. today. And yeah, I'm very happy about this talk. I'm very happy to listen to what you've been doing. We've been using some of his work. We cited some of his works in our papers, and we found his papers really interesting, and they gave us really good hints to apply Lyme for histopathology. So I'm very curious. I leave you the floor, Ian, and please.
Thank you so much. And thank you both, you, Professor Henning, as well, for letting me speak today. So yeah, I just thought, I'd talk about what I've been doing. So this is kind of a mishmash of what I've been doing for the past couple of years, with some ideas and basically some open-ended questions, I guess, about explainable AI in general that I think are worth addressing. So we're going to be focusing mostly about Lyme, but there's other techniques that I'm going to mention as well. So I know that Mara and Professor Henning, some other people in your group are working with explainable AI, so maybe some of the things I'm about to say are already known to you, right? Maybe you're already familiar. I'm going to go over them just in case. If it's too obvious or something like this, you can stop me. I'll just go over these things just in case, though. Then we're going to talk about the data set, the kind of main data set that I used for doing these tests. It's the patch chameleon data set. We're going to discuss Lyme and why one might choose Lyme. Then we're going to discuss a technique that my group developed called EvEx, and why did we do this? Why did we do it the way we did? And then we're going to talk about some results and conclusions. There's some other things in there, but that's kind of the main overview for anyone that's joining in just now. So the basic concept of explainable AI for our problem here, which is an image classification problem, right? It's usually a multi-class image classification problem, is that we have an image. In our case, it's some patch of histopathology, like whole slide images or something like this. We want to know if
sorry
there's
to interrupt you. Yeah,
yes,
sorry. I think we are kind of stuck on the first slide.
Oh.
It doesn't go forward. So I think, yeah, I had the feeling that you were already okay. Good.
Okay. So let me.
I don't know if it's a problem on the screen sharing. Or... Yes.
Now it's
yeah. Maybe I have to. Let me. Try
now we see the first.
You see the first slide, yeah?
No, we don't see anything.
Try this then. This should work a little better. Yeah, it's always tricky with these like screen sharings if you have to choose the full screen or not. Okay, so now you see slide 2?
Yes.
Perfect. So yeah, this was mainly the slide that I was showing while I was talking about, we're going to talk about explainable AI, patch chameleon. Oh, can you see my pointer?
Yes, we can see the pointer, yeah.
Perfect, that's going to be useful. So yeah, when we're talking about explainable AI in general, we have, for instance, some whole slide image. We cut it into patches, and then we're going to have some image like this. We pass it through some kind of model, for instance, a convolutional neural network, one of these things, and it outputs a probability, right? So let's say 0.87, numbers closer to 1 mean tumor, numbers closer to 0 mean healthy. And this, although the number gives us some idea, maybe, how confident, quote unquote, the network was in making that prediction, it doesn't tell us much more than that. And this is why this type of model is called a black box model, because we don't know the internals. We don't know exactly what happened here to cause the network to say this image is a 0.87, right? even if we could open this black box, quote unquote, by just looking at the weights and everything and the activations, but they don't mean anything to us, right? We know that the network learned some kind of representation of the data that's allowing it to classify it, and classify it at a very high accuracy most of the time. But we don't know what that means. It doesn't, looking at those numbers doesn't mean anything to us. So then what explainable AI proposes is we take that representation, or we take, we probe that representation that the network learned in some way that is more meaningful to us, to the humans using it, right? So for the case of image classifications, the most usual approach is to try to generate heat maps, like this one, for instance, where we say, well, this region in the middle was the most relevant one. So this heat map here, for instance, was generated using EvEx. I'm going to explain how we did that. There's a number of XAI techniques that were developed, dozens, if not hundreds. Most of them fall into some categories, right? So some explainable AI techniques try perturbations on the image and then see how that affects the output. Some other ones are more specific to the type of model. So you're using a convolutional neural network, you're going to look at the last convolutional activation, and then you're going to calculate some gradients or something like this. There's two big reviews by Guidotti and Arrieta. And then the importance of these techniques for medicine was stated over and over and over the past years, right? So pretty soon we're going to have these things, if we don't have them already, right? We have these models making decisions in hospitals and things like this. We need them to be a bit more probable, right? This is we can probe them to understand how these decisions were made, especially for these kinds of critical situations, right? And although this was stated very clearly, at the point that I started studying this at the start of my PhD, there weren't a whole lot of papers on this topic, right? People were expressing the need for this, but the applications weren't done yet, or very few. So in order to approach this, we should choose a good data set first, and we'll see, maybe what constitutes a good data set. We must also choose a good technique for the purpose of images, because there's, as I said, there's dozens, if not hundreds of techniques. Not all of them apply to image classification problems, and not all of the techniques that apply to image classification problems are equally consistent or pass sanity checks. Mara has some really interesting work on this, and there are some other papers as well that try to check whether these explainable AI techniques are going to keep working the same if you randomize the network weights, if you randomize the labels of the images, and all of these things, right? So not all techniques pass those tests. As for the data set, there's a bunch of data sets nowadays, open data sets of medical images. We decided to start with Patch Chameleon because it's a pretty lightweight data set in a way. It's easy to work with. The images are 96 by 96 pixels. These are generated from another data set called Chameleon 16. It was one of those big challenges in segmentation of metastatic tissue in lymph nodes, right? So the original images were gigantic, kind of 20,000 pixels by 10,000 pixels or something like this. Wheeling et al., the authors of this data set, they cut it into patches to make it a little easier to work with. There's about 200,000 patches in this data set, and it's roughly divided 50-50 for two classes, healthy or tumor. And so this ease of work was one of the factors, but another important factor for us to choose this was that it includes the medical segmentations. So there were doctors, there were medical professionals looking at this and going, this is where the tumor is, right? And that was very interesting to compare later to see if the network is going to coincide with this or not, right? If whatever heat map we generate is going to highlight the same regions as the doctors did. So that's why we chose that data set. As for why we chose Lyme, so one of the important things here is we didn't choose Lime because it's going to be the only technique that we're going to use, and that's that. We chose Lime first, right? And we want to study all the other techniques as well, and we have studied some of these other ones. But we started with Lime because, one, it's model agnostic, and this is in the name of the technique, right? Local interpretable model agnostic explanations. What does that mean? There's all kinds of classification systems that you could be using. You could be using all kinds of neural networks. You could be using things that aren't neural networks, boosted trees, random forests, SVMs, whatever, right? You could be extracting features from the image using like handcrafted algorithms from the a couple decades ago, and passing those through. There's all kinds of things that you could be doing, ensembles and whatnot. Lime doesn't care about that. Lime only cares that you have a function that receives an image and outputs softmax probabilities. As long as you have that, right, you can use Lime. And we thought this is good because if people want to reproduce our work or do, use it for their models, they could just use Lime as well, right? So it makes sense to start with this one. Now, there's a term here that this is not some kind of official scientific term, but I've been using it. Besides being model agnostic, it's also implementation agnostic. So what does that mean? Every now and then you're going to see a technique that is model agnostic, but it was written, or theoretically it's model agnostic. But if you look at the implementation on GitHub, it's like the code has been done specifically for one type of neural network architecture and PyTorch. And if you want to do that for another one, it's going to take a good while for you to like redevelop the code and whatnot. With Lime, that is not the case, right? It's pretty easy to use it for PyTorch, for TensorFlow, whatever you're used to. So that was also another way we thought it's going to be easier for people to reproduce these results and try to do it themselves and whatnot. So another reason to start with Lime. And also Lime is a surrogate model technique. So what that means is, I'm going to discuss this in more detail, but basically what this means is Lime is going to use a secondary model, a linear model, and linear models are easier to interpret in general, right? You can just see you have several variables, and for each of them, did this one contribute positively or negatively? What was the weight for each of these? It's easier to understand, right? It's using this to approximate the behavior of the more complex model locally. There is an example of this on the next slides. So those are the pros of using Lime. What are the cons? Because all of these techniques, explainable, these XAI techniques, all of these have pros and cons. So some of the cons of Lime, which are cons in a sense also of other perturbation techniques, we're going to see that Lime is a perturbation technique. You have to choose how to perturb the image. And perturbation techniques are only as good as your perturbations are good. So in the case of Lime, it perturbs these images using superpixels. I'm going to explain what these are, but basically imagine an image is divided into pixels. If you group these pixels by similarity of color, similarity of texture, these kinds of contextual informations that make one region of the image separate from another one, different tissues, different whatever, right? You could call these regions superpixels. So Lime, the first step in using Lime is dividing the image into superpixels. You have to choose good superpixels to get a good result with Lime, but what is a good superpixel? So that is one of the main issues, right? There's also the issue of instability. We have some slides discussing this a little bit more. Instability basically means that if you run Lime several times for the same image, you might get different answers, right? And you'd theoretically like to always get the same answer, right? And also Lime is coarse compared to, say, other types of saliency heat maps that might give you, for each pixel individually, instead of using these blocky, chunky superpixels, you get for each pixel whether it contributed for or against the classification, which gives you more detail. But, all of the techniques that give you all of these things, they might have other cons and pros. And some of the techniques that give you a lot of detail, they don't pass those sanity checks that I was talking about and that Mata works with, right? How does Lime typically work? You take an image. So the first step is dividing it into superpixels. You're going to use some kind of algorithm for this. Once you have those superpixels, you're going to generate a neighborhood of around this image of perturbed images, right? You're going to randomly cover these superpixels, and this is a stochastic process. Where you're going to generate, let's say, 10,000 images or whatever, usually like a large number, to create some kind of representative sample. And then for each of these images are going to be sent to your classification model, your neural network, convolutional neural network, whatever it is. You're going to get a probability of, the softmax probabilities. Is it class 0, class 1, with which probability? And then using these informations, you can train a linear model that will tell you for each of these, in this linear model, the weights for each of the variables, and the variables are the superpixels, right? The weight for each of them is the contribution that this superpixel gave in favor or against the classification. So for this type of visualization here, this type of heat map, blue means that it contributed for, red means that it contributed against. In this image, there's no red, but yeah, that's the basic idea. So one of the things with Lime is that it's very common in explainable AI literature. In general, you see it being used more and more. And the two most cited techniques for explainable AI are Lime and GradCam. There's other ones that are being used more and more, but it's usually like the most cited ones are Lime and GradCam. So the problem is when people use Lime, oftentimes they just say, this explanation was generated with Lime, and that's it, right? Lime has a bunch of parameters in it, and we were just talking about how choosing the superpixels is important. So it's interesting to know what segmentation function was used. Segmentation function is the one that generates superpixels. So which one was used? What were the parameters, right? Also, sometimes the heat map is plotted, the Lime heat map is plotted, but you don't get the range of the explanation weights. You don't get those colors with the corresponding numbers on the side. You're going to see how that might be a little tricky. And yeah, does all of this really matter? I think it does, and I'm going to explain why. So this here is an image from the Lime repository. This is the example that they give you, for using Keras, if I'm not mistaken. So you have a cat and a rat, and you classified correctly as cat, and you want to know what superpixels were most relevant for cat. So the example shows this, right? And then I thought the heat maps that I've been using, right? I've been trying to use a color bar and one out of the side. The difference here is, that these pixels were relevant, right? And that this was relevant against. So what it means is this area of the image makes the neural network think that there's something here that is not cat. That's basically it, right? If we were to generate the explanation for the class rat, most likely this would be showing up as green, right? All we know from this is that these regions were relevant, but we don't know how relevant. We don't know if the cat's face was more relevant than the grass or not. And I've seen many papers complain about this, not with this image in particular, but for instance, a complaint of the sort of, Lime is saying that the grass is relevant for the cat classification, so that's not good. But if we generated the heat map differently, we'd see that the grass, although it was positive, it was not as positive as the cat's face. It was actually much less, almost zero, right? So it was positive, but almost zero. So maybe we could ignore that part, right? Maybe, or even if we don't ignore it, we know that it wasn't that relevant. And what we're going to see a little later in this presentation is that much of the instability of Lime is usually related to these regions that don't explain a lot, these regions of low weight. The regions of high weight tend to be more consistent. So just some examples of these visualizations that I think could be more helpful rather than the out-of-the-box one. Because, yeah, when you use Lime just following the example, it's just like that, right? So, and I've seen many papers that use this. And it's tricky to interpret this, right? I think it's easier to interpret it like that. There's other possible visualizations as well, but yeah, so some examples that we've been using in our papers. This one is the one we're seeing where I just use the same kind of interval for everyone, right? Let's say -1 to 1. And this one is interesting in the sense that here I gave you an example where you have an image that was classified with a convolutional neural network. I divided it into 2 superpixels, just one big one in the middle and kind of the background. I want to know, was the middle or the background most relevant? And here we see that the background was more relevant. But then if I randomize the weights of the neural network, I want to see that the network kind of unlearned this, and it shouldn't be able to explain anything. And indeed, it doesn't, right? The explanation weights go way down. The same images, these are the same images, right? And these are more similar to this. This is why I left the cat here. In this case, the same images, instead of making it -1 to 1 for all images, I just decided to go from -max to max, where max is the maximum value achieved in any of the superpixels in the image. It's important to use a symmetrical heat map just to make it easier to interpret the image, because if you don't use a symmetrical heat map, it can be a little tricky. You might see a region as red, thinking it's contributing against, but it isn't, right? So symmetrical heat maps make it a little easier to interpret. And then you just write as the title or whatever what the maximum is. So if we didn't have any of these numbers here, we might be tempted to look at these images and think like, well, this didn't change anything, right? The background is still the most relevant. They're both kind of equally deep blue, right? But no, in this case, the deep blue means 0.05, and in this case means 0.98, right? Maybe to increment this even further, and I'm not sure why I didn't do that here, but we could also add the softmax probability, right, of the classification, which is also an interesting metric to have. On the other hand, if I were to do these images like this, right, like that example, all we know is that this back part here was relevant in favor, and this one, let's say it was relevant against. But we don't know much else, right? And it might be very confusing to look at these and interpret them. It makes it much harder. So on that sense, it's kind of curious that this is the most commonly used visualization. And one of the things is, all of this that I've been talking about, I think that this doesn't apply only to Lyme, right? Many XAI techniques have some kind of step before you're generating the heat map where you either normalize the values before you plot them as a heat map, let's say normalize them between 0 and 1 or -1 and 1, or you delete all the negative weights, you only plot the positive ones. Couldn't we be losing some kind of relevant information in that process, right? Couldn't we be entering a situation like this where we're comparing 2 different heat maps? It looks like they're the same, but they aren't, just because, they look the same because we normalize them to the same range or something, but they aren't, right? So this is actually, I believe, an open question. Many other techniques, we might be interpreting them, and we might be interpreting them differently if we had interpreting them a certain way, I mean. We might interpret them differently if we hadn't normalized them or plotted them a little bit differently. So, okay, with that in mind, I just wanted to make a comment on that. In 2019, we decided to use Lyme to explain classifications on the patch chameleon dataset. We used the 3 different segmentation algorithms that come on Lyme to generate superpixels. And we noticed that it seemed like the FHA algorithms, the Felsen-Swalb-Huttenlocher, this FHA algorithm was generating the largest superpixels that still kind of coincided with the medical segmentation. The medical segmentation is this kind of shadow, green shadow, right? And the explanation weights for FHA were also usually higher than the ones found by QuickShift or Slick. We also tried a different technique because we noticed that it was pretty hard and arbitrary to define which parameters we're going to use for the segmentation. So in this case, we're just kind of trying to obtain the same number of superpixels by manually tuning these parameters. Because we want to compare, well, if these 3 are going to separate the image into, let's say, 30 superpixels, which of them finds the best 30 superpixels or the 30 superpixels that most match the doctor's segmentations or something, right? What if we use a technique that doesn't have all these parameters to tune? What if we just divided this image into a bunch of squares and we kind of overlapped several of these square grids of different sizes? Of course, it would show a rougher kind of heat map, but it doesn't have any parameters to tune. So maybe we should try that, right? So that's what we did, divided the image into a bunch of square grids. Each of those is passed to Lime as if it was the superpixels. I mean, these are not technically superpixels. They don't hold contextual information and etc., but as if it was, right? And then generate the explanations with that. And then just merge all of them into a final heat map. And what we saw is that they seem to agree a lot with the other more sophisticated segmentation algorithms, right? So that was an option that we were keeping in mind. But what if there was a way for us to kind of not be manually and arbitrarily, or as arbitrarily fine-tuning these parameters, right? So what we decided was, since we realized that FHA was finding these largest and these largest superpixels with higher explanation weights, why don't we use that in our favor, right? And try to use an optimization algorithm that just maximizes that. So let's say I'll use a genetic algorithm that tries to find the parameters for FHA that generate the largest weight on the best kind of the most explanatory superpixel. The problem with that approach is that as soon as you do that, you say, find me the largest kind of weight on a superpixel you can. The algorithm immediately goes kind of, okay, I'm just going to cover the entire image in one superpixel, right? It's just a gigantic square that covers the entire image. And of course, if you ask which part of the image is most relevant for the classification, well, just cover the entire image, The entire image is relevant, right? If you cover everything, there's something relevant in there. So we had to think, well, okay, maybe we want to maximize the explanatory weight, but we want to also at the same time minimize the area. So this already complicates the problem a little. Now it's a multi-objective optimization, but we decided to try it. And that's what we did. Basically, we were using FHA, which has this tendency of finding these big superpixels. Let's do that while also trying to minimize the area, right? And this solves some of those instabilities or those problems, those cons that we were talking about with Lime. So how do we choose the best superpixels? In this case, we let the GA do this according to the metrics that we defined. The instability, okay, so one key thing, we're going to talk about this on the next slide, but a multi-objective algorithm like that doesn't have one single best explanation or one single best result, right? A multi-objective algorithm like that is going to form several explanations that are kind of as good as each other or the best that they could be with a trade-off, This one has a little bit of a larger area. This one has a little bit smaller area, but larger explanation weight. You could kind of give and take, right? And the name of these best options, they form kind of a front on this kind of space of solutions, is the Pareto Front. Because this situation where you're trying to optimize one thing too much and then you start losing on the other one is called Pareto optimality or Pareto efficiency. There's some names like that. So if we take the Pareto front of this multi-objective optimization, then average them, we see that the areas with larger explanation weights are less unstable than the ones with smaller ones. And this approach in general is less unstable than Lime, kind of pure Lime, right? Averaging the best explanations found by this process is less unstable than Lime. The coarseness of Lime is also solved a little bit by EvEx because the Pareto Front averaging makes the final heat map a little bit more detailed. But of course, this is still way more coarse than those saliency heat maps that give you pixel by pixel. Though, again, there's give and take, and we're going to get more into that as well. So a quick note on instability. Where does instability come from in Lime? So there's basically three parts to it. The first one is, and let me just make sure because everything is so silent. We're still working. Everything is working. Audio, video. Just making double sure.
We're following.
Perfect. Okay. So 3 sources of Lyme instability, right, that we noticed when we were running this. First one comes from the randomization within those segmentation algorithms. Most of them have some kind of random process internal to them. So that is one. The other one comes from Lyme itself. Remember, we have to generate those perturbed images. So that is a stochastic process. On this run, you generated that perturbation. On this one, generated different ones. You're going to get slightly different explanation weights at the end. Third one, which was the least obvious one, is from CUDA. So when you use a GPU to run your neural network on Keras, on PyTorch, if you're using the GPU to accelerate the calculations, that causes some instability on the results as well. Now, the thing with these is, I think I kind of mismatched a little bit the order of the slides here. I'm going to skip ahead and then go back. How could you solve those instabilities, right? So for the randomization within the segmentation algorithms, let's say we use a seed, right? And the same thing for the stochastic process of generating those images, the perturbed images. We use a seed for both of these just to solve this problem initially. What we realized then, this is why we noticed the results were still varying a little, even with random seeds, because of this GPU issue. So we didn't really find a solution for this besides not using a GPU. Of course, that's not practical because that makes the calculations much slower, right? So what are actual solutions? Because these are just kind of solutions, right? And even if you use a seed, the next time you run a different seed, you're still going to get something different. So those are not really solutions. EvEx helps a little with this because you use a seed, to test one specific kind of parameter combination, etc., for this seed, but different seeds should agree, right? Or should reasonably agree on the most relevant areas. And you can also use the GPU because the instability generated due to CUDA is quite small. It's usually, many decimal places, kind of 0, 0, 0, 0, 0. So usually that's fine. You don't have to worry about that too much. And that's only going to change something more relevant, I guess, for areas of low explanation weights. And that's why the visualizations are so important, right? That whole discussion that we were having. Here I just put an example. I guess I misplaced this slide just to show what a Pareto Front is, right? So this is from the EvEx paper. If you generate a bunch of explanations, each symbol here in this plot is an explanation, evaluated according to 3 metrics, right? The first one is the explanation score. In Lyme, the explanation score is the R² of the linear regression used to do that surrogate model. Largest weight is the weight of the superpixel with the largest weight, basically. So what weight that was, right? That's the largest weight. And then relative area is the area of the superpixel of largest weight divided by the area of the image. So the orange X's here are the initial population of explanations we had. We just generated a bunch of random, FHA parameters, just segmented it randomly, and generated a bunch of explanations. And then we used the genetic algorithm, and you see that they start pushing the population towards this front here. This is the Pareto Front that I was talking about. In this case, due to some technicalities of the library I was using, it only accepts minimization problems. So I wanted to minimize the relative area, so that's fine. It's just the relative area. But I wanted to maximize the explanation score and the largest weight. So in order to turn this into a minimization problem, you have to do 1 minus explanation score, 1 minus largest weight. But you see that all of these are being pushed towards 0, but it forms a front, right? And the idea is that none of these is the best, exactly. We could choose any of these as the best, depending on what we think is more important. For this particular one, is the relative area most important? Okay, so I'm going to go with that one. Or is the explanation score more important? So I'm going to go with something here, what we did instead was we took all of the things that are all of these explanations on the Pareto front and averaged them into the final explanation, right? So, okay, what does the average explanation look like with EvEx? We took some more images from Patch Chameleon. So say here's an image, here's the medical segmentation. So here's the EvEx explanations for 4 different random seeds, right? If you did this on Lyme, pure Lyme, these would vary much more than they are varying here, right? And most notably, so here's some other examples as well, right? We see that they're still contained usually inside of the medical, or even this one goes outside a bit, but the regions that are darker, the most relevant regions, are usually agreeing with the medical segmentation, right? And the difference, one of the most important differences here from this work to the previous one is that there was no manual fine-tuning here. It wasn't us trying to fine-tune the results, the parameters to get this result of, yeah, this matches the medical thing. It's like, okay, we use this optimization algorithm with these objectives, and that seems to generate explanations that agree with the medical segmentations. And here we see if I took these 4 seeds, for instance, and I take the standard deviation of these 4, right? So for each pixel, how much it's varying, we get a plot like this. And what this plot shows, if we compare it to the previous ones, we see that there are some areas, and it's a little bit easier maybe to look at the relative standard deviation in a way. But basically, like, so these are the areas of highest standard deviation, right? But this is a little tricky because this standard deviation, we should be maybe comparing it to the average value of that pixel, right? Because for a pixel that has a very low explanation weight, a variation that we might perceive as small here, close to 0, might be 300% of the value of that weight, you know? For that particular pixel, it varied a lot, even if for us it doesn't look a lot like it. So we looked at the relative standard deviation as well. We see that there are some regions here with very high relative standard deviation, but these coincide in this previous image to regions that have very low explanation weight. So this is exactly what was like happening. The regions that vary the most are the ones that have the lowest weights. So let's say something here has a weight of 0.0001. If it changes to 0.0002, that's a 100% change, right? But it's a 100% change in a region that doesn't explain much, and it's not very relevant. The regions with very high explanatory weights have very low variation, right? If we, for instance, say, you know what, we're going to generate an explanation with Lyme or EvEx, but we're only going to pay attention to explanation weights above 0.5. And 0.5 is a pretty low cutoff, but let's say like 0.5. We see that a lot of the regions of high variation or high relative variation are eliminated, right? So on that sense, there is instability, but there's less instability if you choose regions of high weight, and if you average the Pareto front, there's even less, right? So that's, you know, Lyme and EvEx. There's other perturbation techniques. One of them is called RISE. Both of these are more recent. So Lyme is from 2016. All of these things are recent. Everything in our area was published, like created like 4 years ago, right? Everything is recent. But meaningful perturbations and RISE, if I'm not mistaken, are from 2018 or 2019. So a little bit, you know, more recent, even more recent. Meaningful perturbations tries to do something similar to Lyme, but instead of using superpixels, it creates this kind of big perturbation and uses gradient descent to train this perturbation to try and find the perturbation that changes the prediction probabilities the most, right? RISE, on the other hand, instead of choosing superpixels, just generates a bunch of random kind of masks, perturbation masks, just basically covers the image randomly in a bunch of different positions. And then it sees how much each of these perturbations changes the prediction probabilities, uses that as weights, and does a weight average of each of the masks multiplied by the change it made on the prediction probabilities, right? There's also gradient techniques, which are very popular. So some of them, GradCam, integrated gradients. There's a bunch of gradient techniques. I'm not going to get into all of these here. the thing is, not all of these pass sanity checks. So what are some of the common sanity checks? And Mara could explain this better than me, but they check, for instance, randomizing the labels, randomizing the network weights. Does this change the explanations or do they remain the same? So integrated gradients generates these beautiful explanations, right? When you look at them, it's like, wow, this fits my intuition so much of what part of the image is important or not. But when you randomize the weights and whatnot, the explanation remains mostly the same. So that's a problem, right? You'd expect the network to lose this kind of information that it had if it's randomized. So they see that some of these techniques, while they generate these very intuitive explanations that seem very good, just by looking at them, they might be just kind of doing edge detection or something like this, which doesn't really explain much, right? Some other applications besides metastasis and metastatic tissue that we are working on are for COVID, right? I have results. I have interesting results even about this. I didn't add them to this presentation because they're not published yet, but we do have some COVID results, both for X-rays and for tomographies. For tomographies, the most interesting aspect is that one thing that we noticed with this is a lot of CNNs and other types of systems that are being trained right now are being trained on very small data sets because it's the reality of COVID data sets right now. There's not a lot of open data on this. Most of them are crawled, right? They do these crawlers that go around picking images from papers. One of the things with this is these data sets are extremely imbalanced and inconsistent. And most notably, the tomography ones have a bunch of artifacts on the images, like letters from the paper, the different papers they crawled, little letters, colored letters. So all of the images are black and white, but some have like a red A on it, like a label from the image. Using these explainable AI techniques, we showed that the networks are not paying attention at all to the body, and they're just paying attention to those like irrelevant artifacts, right? So that's an interesting thing. And also a malaria one. I put some quick examples, but one of the things with these black and white ones is that we might not be using the same perturbations we would in Lyme, right? If I just start covering this in random black superpixels, maybe this is going to affect the explanations in some way. Maybe we're introducing some biases because this contrast of black and white here is important, right? So maybe we want to do some different types of perturbations. Lyme doesn't offer these, so we had to implement them. But basically, like maybe we want to perturb this with a Gaussian blur or with some other type of noise, or maybe some other ones. So we started by doing this Gaussian blur, Gaussian noise, and whatnot. But maybe there's other types of perturbations we want to look for, and how these perturbations maybe affect the explanations, right? I just put a quick example here also of a malaria data set. So malaria, we have these kinds of two images from the data set, the common blood cells and the blood cells infected that show these purple kind of blobs, right? We trained the ResNet 50, using some kind of common transfer learning from ImageNet, like the typical approach. And we used some heuristics. We wanted to test if we could use some heuristics for FHA and then compare that to Squaregrid. So if you take an image and you just divide it with FHA with standard parameters, it finds all of these kinds of superpixels. But what if we wanted to say like, and that's one of the advantages of perturbation techniques, especially with Lyme, because it's usually seen as a disadvantage, but I think it could also be seen as something interesting. If we want to say, let's say compare this region to the cytoplasm, for instance. Which of the two is most relevant? In most techniques, you don't really ask, is this more relevant than that? You just ask, what is more relevant? In Lyme, you can kind of ask, like, is this more relevant than that? Is this region more relevant than that? By just segmenting it like that, So we saw that if we use FHA with the scale parameter of 50, it typically segments the purple blob from the cytoplasm. So that creates an interesting question, in our opinion, right? How does this purple blob compare to the cytoplasm? And now we can generate a Lyme explanation for that. And the result was that in general, the purple blob is more relevant than the cytoplasm, right? The cytoplasm might contribute for or against, but usually the weights are so low. And from what we see, it's a little dangerous to be interpreting too much of these low weight regions. The highest weight regions in these images are usually the purple blobs, which seems to make sense, right? But also using Squaregrid, which didn't have any of those parameters, we also highlight the same purple regions, right? So, and here is just an example of another visualization that I hadn't shown before, where you just, you kind of create a boundary around the region of a certain threshold of explanation weight, right? In this case, let's say 0.2. Then you can kind of mark these regions a little bit more easily on the original image, right? So some open questions that remain. These different techniques, the different XAI techniques, a gradient technique that is asking the question, if I calculate the gradient from the last layer or the output layer to the input layer, right? What was the most, the highest, what was the pixel with the highest gradient? So that's one question. Another question is, if I divide the image into superpixels and I train a surrogate linear model, which of these superpixels has most relevance for the linear model? These are two completely different questions. Yet it's pretty common to see when you're going to look at these XAI papers, they're comparing all of these images side to side, like LIME, GradCam, integrated gradients, whatever other techniques that they're using, right? But these things are not asking the same questions. So should we be expecting the same answers from them, right? If I generate a heat map with LIME and with RISE, should I really expect these to highlight exactly the same things? Maybe not, right? Maybe the different questions will highlight different things, and maybe that's not bad, right? Maybe we need to use all of these techniques together to try and understand the most we can about a model and about what a model is doing. But also, are any of these things that we are generating what the CNN knows, right? Because that's the idea, right? How do we know that this is what it really learned? Maybe we don't, right? It's almost verging on the realm of philosophy of what the explanation means or what it really knows. But maybe we don't know that, and maybe we can't know that. But we can extract useful information. We can say sentences like, if you perturb this part of the image, the probability, the prediction probabilities change by that much. That's something we could say with relative certainty about a given network. Whether this is what it knows or not, that's more complicated. But we could say, the gradient of this area is, this area, usually this type of texture has higher gradients. How can we use this information to trust more or less what this model is saying, But these are some open questions. I don't think they're answered yet. And I hope that all of us can answer them, right? So some current and future directions include some perturbation techniques require finding good, or all of them actually, require finding good perturbations. So we need to continue defining ways of how do we decide what a good perturbation is, because perturbation techniques have this promise of, or this advantage, I guess, of you being able to say, I want to see what that part does, let's perturb that part. I want to see what that does. But we need to find some more standardized ways, some standardized approaches to doing this. A lot of gradient techniques, although they generate those beautiful heat maps with like all the details pixel by pixel, they don't pass sanity checks. And there might be sanity checks we haven't developed yet, and some techniques that are passing all of these sanity checks, maybe they're not going to pass future sanity checks that we come up with, right? There isn't a standardized approach to evaluating this explanation yet. And also, one of the things I think we could do, and we should do, and I want to do, right? Maybe on a postdoc or something. I'm about to finish my PhD now, so I'm thinking about all these things. How can we create a data set that like an artificial data set that challenges these explanation techniques in specific ways, right? So we have because one of the things we have is that we're using this metastatic tissue data set, but it's kind of hard to interpret exactly what explanation we're going to look for, what exactly is the right answer for this. Even if the doctor marks in a region says this is the metastatic tissue, but that's still kind of hard to interpret on a more kind of minute way, right? Maybe we could generate a data set, an artificial one, where we know exactly what we're expecting, we know exactly that the classification has to highlight exactly this area in an explanation or something like this. Otherwise, the network is not focusing on what we know we want. So there could be some standardized data sets to test XAI. I think that's a possible future direction. Here's just some things that we implemented. So the Squaregrid implementation, if any of you wants to try it, I put it on GitHub. It should be pretty easy to use. You can ask me anything. And I also made one for Rise, that other perturbation technique that generates the explanations by perturbing things with random masks and then doing a weighted average of them. I also put like a TensorFlow implementation of this on GitHub. And yep, that's all we had for today. I hope something was useful.
Thank you very much, Ian. It was really interesting.
Thank you.
I would like to see if anyone else has questions. And I noted down some of mine, but I will first ask the others if they have questions. So,
thank you very much. It was very, very interesting. So I have a couple of questions. So of course there is a trade-off between the finer the grid, the better explanation. Well, it depends on the task, but you could expect that is more fine-grained the explanation. But then the problem is that these whole slide images are huge, as you said at some point, right? So is good and in this case scenario where you're analyzing just image regions, but what happens if you have to give an explanation for a whole slide image at test time that has probably 10,000 or 20,000 of these patches? No, maybe I'm exaggerating, maybe 1,000 or 2,000, but in the order of 1,000. So that do you see some sort of way to escalate these techniques like for that particular task?
Yeah, so that is actually one of the biggest issues to tackle. I forgot to mention that actually. One of the cons of perturbation techniques is that you have to be calling your CNN all the time, and that increases computational cost a lot. Exactly, right? So they are slower. That hasn't been solved yet. So for EvEx, for instance, we generated those interesting explanations, but it takes 8 hours on CPU. 8 hours to generate an explanation for one patch. So to explain a whole image would take a very long time. Although, we weren't using any kind of supercomputer for that. So maybe with more horsepower, it would be faster. Also, GPU usage cut the time by a lot, right? It went from 8 hours to 20 minutes or something, even on our weak machine. So maybe that, but also I think that as we develop faster and faster implementations for these things, that's where that thing comes in, where we got to combine these techniques, I think, right? So gradient techniques generate explanations almost instantly. They're very, very fast. So maybe we could start combining these things. Maybe the regions of the image that have the highest gradients, so maybe then we go to those and do a perturbation or something like that. So something of this sort. But that hasn't been solved yet. These perturbation techniques really don't escalate well. Your question is completely pertinent.
Okay, so it's still, but that's good. So there is still some problems to solve. So
there are.
I would say there are many problems.
There's many problems to solve. That's true. That's exactly true. Yeah.
There is another one that I annotated. So the method favorizes the explanation with large areas, right? So which is one of the objectives of the SGA, the genetic algorithm. But there could be some tasks where the explanation should be small areas that many like they're uniformly distributed across the image. So, for example, nuclei that have to be equally relevant. So do you think that this one will be catched by the Pareto frontier, or they should? Right?
So one of the things that I didn't mention here, but EvEx, like the idea is that you could adapt this to different problems, right? You don't have to use Fels and Swalb, for instance. You don't have to use that segmentation. You could use a different one, and use the same kind of evolutionary process for a different set of parameters for a different set of segmentation functions. And in the same way, you could also use different goals. So if you're going to use an image that you know has multiple relevant areas, maybe you could change the goals a little. Instead of maximizing one kind of superpixel, maybe we could maximize 2 superpixels or something. Like the 2 most relevant superpixels, or 3 most relevant, or and most relevant. But then we're going to have to come up with more complex goal functions. But yeah, it wouldn't. Probably if we applied exactly this how we did it, it would have some problems, but we could change the goal functions to adapt to this problem. It's adaptable.
Okay, cool. And the last one was, I think that does everything. So there is this idea of just masking and randomizing and having and doing the prediction, coming back, averaging. This is some sort of reminds me a lot what is happening now with these transformers architectures, where you just mask the input, have many they have outputs that are relevant for subsequences of the input. Mm-hmm. So because I have seen some recent papers that on this unexplainable that they use transformer models to generate counterfactors using these combinations of the sequences of the input. So why don't just transform? Because at the end you are also trying to find a combination of these parameters that maximizes your explanation. Don't just put a nonlinear layer on top, which is basically what transformers do. So basically they are replacing the genetic algorithm by another neural network, which is just make the thing more complex, but maybe that also make some sense for some more sophisticated explanations.
For instance, I wasn't aware that they're using transformer models to classify images now as well. I didn't know. Oh, that's interesting. I have to look into that. That's pretty cool. I didn't know. And that technique, I forget the name of the author. We're talking about Rise, right? The one where you randomly kind of cover, and I think it's Petsyuk, the name of the author, Petsyuk and his peers. I can give you a link to the paper. I'll send you something. Yeah. We can try it. I think your suggestion is completely valid. We should try it. The code is pretty easy to change. I'll send you the link to GitHub. It's pretty easy to adapt. Should be able to put a layer, a linear layer, a nonlinear layer. Sorry. Yeah. Thank you so much for the suggestion, by the way. Yeah, very interesting.
Thanks.
Mm-hmm.
Are there other questions? So while the other things, maybe I will ask some of mine. Sure. So at the beginning, you mentioned about implementation agnostic methods. So you discussed, in fact, that it's something that it's kind of a reality for us that's working on explainability techniques. Mm-hmm. That if you say, okay, I want to compare this method to another method, and you go on GitHub, and it's like, yeah, that's great. There's a GitHub, and oh no, it works only for PyTorch, it works only for TensorFlow, it works only for whatever. So I was wondering, like, do you know what would be the impact of this approach that we have on research of using only this method, this implementation, on interpretability research? So what is the impact of having one technique that doesn't necessarily talk to other tools?
I think that all techniques should strive to be as usable as possible in all different platforms, because one of the things that is not clear, I was trying, for instance, to recreate the meaningful perturbations, right? That's one of the techniques. I was trying to recreate it on TensorFlow, and I just couldn't be sure that I was doing the right thing, because the way that you calculate gradients in TensorFlow, the way that you calculate gradients in PyTorch, there's a ton of intricacies into that. There's a ton of little parameters that you could change here and there. And I'm not sure if I'm doing the same thing, right? So I think that on that sense, we might be generating heat maps that could have different things. And the thing with, for instance, with the meaningful perturbations, you have to actually train a thing by gradient descent, right? So you're training that perturbation with gradient descent. Your parameters for that gradient descent affect the final explanation. And I wasn't generating good explanations in the end. They had all these artifacts, and I'm sure it was I was doing something wrong, but it wasn't easy to figure out what, right? So I definitely think on that sense that, and the thing is, let's say that I didn't know this. Let's say that I didn't know there's something wrong because I never saw the other heat maps from the other implementation. And then I put those in my paper. I say, like, these are the heat maps. Meaningful perturbations
exactly.
has all of these artifacts. Maybe it doesn't. Maybe it was something on my end and I didn't even know. So yeah, like, I think it's important to try to unify these as much as possible. And yeah, makes total sense. I think it's a great question.
Yeah, I think it's practicity and usability of the tools that we develop. It's something that we need to take into account when we propose something, because this might be used also by other people that have a different target. So
yeah, of course,
I think,
yeah, like a doctor might not know what a superpixel, a medical doctor, I mean, like, he doesn't know what a superpixel is, what gradient descent is, and it should be as easy as possible for them to use it, I think.
Yeah, that's sort of what I think too. And there are I know that there are some students with us, following us, that maybe are more on the pathology side. So if they have comments, I would really invite them to help us with this, because I think sometimes it's difficult to communicate between the two worlds. And one of the reasons why we compare our explanation, or the explanations that we managed to generate at the end with the doctor's annotations, it's because we don't know exactly what we would like to have as an explanation. Because if you were to ask me, I would tell you, well, I would have probably to study many more years and something different from what I've done to be sure that this explanation is actually enough satisfying, as I think it is. So yeah, that's sort of I think it's a big wall that, especially when you apply explainability in the medical domain, it's kind of different from natural images, where more or less all of us kind of have a common ground that is solid and shared.
This is a bird. Yeah, like, yeah. We all understand what a bird, what a tree is, but then metastatic tissue. I completely agree, and we had so many plans of doing collaborations with kind of medical research teams and whatnot, but then COVID happened, and all of them are completely busy now, and it's not really happening. But I think that's kind of the next step, right? Now that all of these explanations, XAI techniques are being generated, kind of integrating it more with the medical part, for sure. Like, absolutely, I agree.
Then I have a question that is maybe more technical and less on the approaches. How do you? So you used Lyme, and this is for the pathology experiments. So you used it on Chameleon 16 because it's the patch Chameleon dataset. So it's nice and easy. I was wondering if you have checked how does it react to domain shifts. So for example, the Chameleon 17 has it has images from 5 different centers. So there were different staining techniques and 3 different scanners. So you have small subtle differences between the images. That you already have this in Chameleon 16, but in Chameleon 17 it's even more. It's more stressed as a difference. And I was wondering, like, if the explainability technique itself should be checked whether it's robust to domain shifts, so to data that come from a different domain. Because this is also what we do with classifiers when you evaluate uncertainty. You don't know if the uncertainty comes from your data or if it comes from the model. So I was wondering, like, if there is some kind of uncertainty in the explanation coming from the explainability technique itself, but also it could come from the data that come from somewhere else.
Yeah, I totally understand what you mean, and I think even on the original Lyme paper, right, by Ribeiro, right, he mentions that he says something like Lyme is not going to be able to explain all kinds of classifications because let's say they have some data set where the relevant factor is whether the image is sepia or not, or some sepia or something like this. You can't cover the entire image, or even if it says, oh, this is the patch that's important or not. You know, right? You're the human. You know that it's the entire image and the kind of the texture, this or that, or some other thing, but you're not going to find that out by doing this process of covering superpixels. So in some cases, that's definitely going to happen. And this is where there's that benefit of combining different approaches. And I think that that's, at some point, I think what's going to happen is there's going to be a suite of explainability techniques, right? And if someone is analyzing an unknown problem where we don't know anything of what might be causing some classification or not, people are going to use 5, 6, 7 different techniques and kind of combine all of them into one big overview, right? I can tell you that one of the data sets that I was studying more recently, so I haven't looked into Chameleon 17. I'm going to look into it now. I'm very curious. I was looking into those COVID CT data sets, and those suffer massively from this. Images have different aspect ratios. They were taking in different things. They were literally just kind of crawled from the papers and resized. So they have very, very different characteristics. And it would be very easy for the network to just learn it's almost easier for it to learn which type of paper it came from, right? Than which disease it has. So
I mean, the problem with that is really also that when you take it from a paper, you don't have the DICOM resolution. I mean, it's just completely different. It's not only things being added, but you don't have the 12-bit gray levels, etc., etc. So you don't know what level window setting they use to save the picture. I mean, I've seen some of these, but there are a couple of data sets. Unfortunately, most of them are not publicly available. And but data sets are being created in institutions, etc. It would just be important to have them available as well. There was a challenge, I think wasn't there a MICI challenge or something on COVID as well, or RSNA? I don't remember, but there were some challenge data, and I would like to look into these actually.
Yeah. That's the main issue right now with these COVID classifiers, is exactly what you mentioned. The best data sets are all private, and the open data sets are usually very tiny and with these crawled images that,
are you talking about the GitHub repository of COVID images, Ian? Because I think I had a look at that, and most of it is crawled images from papers, publications, and
I mean, it's also people discussing on Twitter, etc. I mean, there's a variety of images that are available, but none of them are original resolution. Often it's screenshots from a viewing station, so you have like a few slices, but you don't have the full stack of images. So it's really
there's like there's 2 or 3 data sets, I think, like 2 on GitHub, 1 on Kaggle or something, and everyone that's doing research with this is kind of mix and matching, combining these 3, like doing augmentations or something. But what we are kind of showing, and I haven't published that yet, but what we're kind of showing is that at least for that CT data set, the network doesn't seem to have learned anything relevant about COVID. It's never paying attention to the chest area. It's just like paying attention to the random letters on the back. It's so clear. There's some examples. Yeah, like the letter A, the letter B or something, and then there's like a gigantic explanation weights just on the letter, and the rest of the image is irrelevant, So it's like we shouldn't.
I actually need. I'm sorry, I need to leave now.
I'm sorry. I went a little
but you can
over
continue.
time. It's my bad.
No worries at all. Yeah. Thanks a lot for the talk. It was really great, very interesting.
Well, very nice to meet you.
Nice meeting you. Bye.
Bye, honey. Yeah. No, it's also that the purpose of interpretability in the first place at the beginning is like when we develop these models is making sure that debugging purpose is the first, I think, the first use case for that. So you also already kind of answered to one of the other questions that I noted down, that was about when you're optimizing the generation of the superpixels with different objectives, and you have this Pareto frontier. Like, one of my first concerns was really computational issues. So you kind of talked already and you touched it already, and I think
it's very slow.
But it's a beginning. It's a beginning. It's just that eventually one should converge to having explanations when they're needed right at hand. But yeah, there could be some high computing or some kind of CPU-based algorithm. It's
Yeah. Like I can tell you, for instance, like there's a group from São Paulo that wanted to collaborate with us, and they're studying melanomas, right? So the images on that data set are quite big. Like the patch chameleon ones are 96 by 96. These ones are, I don't know, 500 by 500. It already took like 20 hours to generate an explanation with EvEx, and that's just not practical. That's not practical. 20 hours on like the CPU, 8 hours with a GPU. It's not practical. So we are trying to find different ways to go around that, right? And this is where I think Squaregrid helps a little bit, because even as rough as Squaregrid is, it's faster than EvEx, right? But it's going to be a combination of what computational resources we have available, how much detail we want in the explanations, right? Like all of these things are going to play a part, I think.
Okay. Are there any other questions? Yeah, I see Sebastian has raised his hand. I don't know if it's
Yes, so I have been thinking in this problem for a quite some time now, because always in our last paper they have this issue that we always say our methods might have overfit to a particular set of annotations from a pathologist. So that's an hypothesis that we have from a long time ago, and that depending on which kind of set of labels or pathologies do the annotations of the data set, you will have certain performance, and of course the performance will depend on that. But what will be really nice, a good use of this set of tools or explainability methods will be to show actually that the explanations that one might get for a certain pathologist differ from the explanations to another pathologist. And I know a couple of data sets where this could be applied. So maybe we can I don't know, maybe we can set up some sort of collaboration or something. Because I think the problem is very is there latent, and there is still not yet some like quantitative tools to show, hey, look, it's actually different, the explanation.
I've seen something like this. I recently was like reviewing a paper on one of these things where without getting into too much detail, but basically they were trying to analyze precisely this, right? Like, are the classifications of this system closer to this pathologist or that one, you know? And they weren't using any of this. Like it was an image classification problem. They weren't using Lime or anything like that. It was like a completely different approach. But we could combine all of these things, I think, right? Like there is like many different ways that this could be tackled. And I definitely think these heat maps could help, right? I definitely think that they could be useful for this.
Right. Yeah, to show like an average opinion, like an average annotations or explanation might be the most reliable. That could be interesting.
Yeah, and I mean, what you said about collaborations, yes, of course, that would be. I really like that idea, and Mara as well. Anyone that's watching, if you think that there are ways that we could apply this or something, collaborate, by all means. I think it's a great idea. I think we're the ones that are gonna be driving this forward, right? Like this kind of explainability discussion and whatnot. Like this is a growing area, and it's mostly being done by PhD students, by all kind of this new generation of researchers, I think.
Yeah, no, we definitely will keep in touch for sure, and especially your Lime, your work on Lime is very interesting to me, because my first impression with it, and I also wrote it in the email to you, I think, was that it's pretty tricky and it has to be taken with the level of expertise that requires to be applied is different from the level of expertise that you think it would require to apply it. So it's really and one thing is understandable, because it's as you were saying, it's promoted, and it is a method that is easy to apply, is intuitive to understand at the end of the story. But then when you look into all these little subtleties of the application domain, where you end up applying it, it becomes, it requires a different level of expertise and many more hours that you've been working on it. So with this hours spent,
that's kind of how it was with, like, at the start of my PhD, we were thinking, okay, let's just apply all of these techniques, right? Like Lime and GradChem and this and that. And then we applied Lime thinking like it's going to be like 10 minutes. And then all of a sudden we're working with Lime for 4 years trying to, like, you know, improve and thing and that and understand this and that. So it was.
I agree.
You know.
Yes. And I think that the next step is going to be to make this actually useful for clinicians or for the end users of this, because from our perspective, when you were saying we're asking different questions, is it really given, and is it so obvious that the answers should be the same? I think at the end of the story, what we want to give is something that doesn't create additional uncertainty on the methods unless necessary. So if it's necessary because the model is not working, then I agree with you. We should point it out in all possible ways, and that's for debugging purposes. But when it's not anymore machine learning experts using the explanations of the model, I think we would like to converge to something that could be an average, could be a stacked approach as you were proposing. So having like first one method and then another method and then a last method, or a combination of stacking and having complementary information displayed. But we should come up, and I think I haven't seen this quite yet, because all of us are a bit focused on, we choose one method and we focus on it, and we live in a little world. And so that's kind of what would be interesting to have, I think, as a step for sure. So we will keep in touch, I think, because we're also working, and I would like to at least understand how to apply your tool, but I'm not so, I don't know, I think 20 hours per explanation would take me too long.
Yeah, depending on your data set, maybe it's not gonna take that long, hopefully. But yeah. We're also maybe you have access to some more powerful machines than the ones we have here. But yeah.
We could try to see if we can scale it up to
I'm also
1,000 of CPUs.
Yeah, I'm curious how it's going to do with other data sets and other settings, if we try different goals, different metrics, all of that, right? Like, all of these tests are important, and I'm curious about them too.
Okay, cool. If there are any other questions, I would say this is the last call, because we already passed 20 minutes the 1 hour. So I would like
I'm so sorry, but
I don't know. It's fine. It's really interesting, and I'm happy that there's plenty of time for questions. But I would like maybe to let you go and to let also the others free. So if there are any other questions, I would say this is the last call. And if not, I think that's it. So thank you very much. It was again very instructive, and it really showed me that there could be a way to apply Lime that is probably more trustworthy, if I'm using the correct terms. So yeah, that was really interesting. Thank you very much.
Thank you so much, Maren. Thank you for the opportunity. I mean, we had like such an interesting conversation, and it led to this presentation. I'm very glad. Thank you so much.
Good. So thank you. Bye. Have a good day, evening. Not sure what time is there.
It's 1 in the afternoon. So I think you guys are reaching evening soon, right? So have a good evening.
Thank you. Bye, everyone. Bye.
Bye. Thank you very much.